So, now let's get into chemical reactions. Chemical reactions is how we change a molecule from one shape to another shape, or how we put atoms together in order to form a molecule. There is a principle that we always have to follow in every chemical reaction, and there's actually a couple of them. The first one, the amount of mass we start with is the amount of mass we end with. Mass never disappears. If we start with 100 grams, we have to end with 100 grams. If we start with 100 atoms, we have to end with 100 atoms. It's what's referred to as the conservation of mass. The other thing we have to remember here is that it's based off of the laws of thermodynamics. And the two key principles we have to remember: energy can never be created, nor can it be destroyed. That's why I said you can't burn a calorie, because a calorie is a unit of energy. The only thing you can do with energy is convert it from one form to another form. And the typical conversion is from a potential form to a kinetic form. Kinetic is the big fancy word for motion. That conversion is never 100%. We always lose energy. Things that go from being big to being small release energy into the environment. Things that go from being small to being big absorb energy from the environment. Anybody play billiards or pool or snookers or any of the other games where you have to smack a ball into a whole bunch of other balls, or bowling? In bowling, when the pins get laid down, or when you play snookers or billiards or pool, when you lay out the rack, the balls have the pins have the maximum amount of potential energy, because they're sitting there and they're waiting to be moved. You then hit the cue ball into your rack, and you hear what? You hear the crack. The crack is the release of energy from the big thing into all of the small things. That's the release of energy. We have the same thing happening within our body in all the metabolic processes. But we don't hear the energy, we feel the energy. And we feel the energy in the form of what? We feel the energy in the form of heat, what we call thermogenesis. Thermogenesis is the release of heat, or the production of heat, from all of the chemical reactions. And we have to have enough thermogenesis in order to keep our body temperature about what? We're in science, and so we don't use Fahrenheit. And I'll give you a story time in a second here about why we don't use Fahrenheit. 37 to 37.27 is about what we want. If we go above 40, no bueno. If we go above 41, anybody watch any marathon running or long distance running or anything like that, where they collapse as soon as they cross the finish line, or they collapse halfway through the run? 90% of the time, their body temperature is somewhere between here and here. Anybody ever exercise or exert themselves so much that they start feeling what they call the stitches in the side? That's when your body temperature is getting close to this level here. So, when we look at chemical reactions, there's a couple of things we have to look at. The first one, there are two key concepts of energy. One we call free energy, even though it's not really free, it costs us a whole bunch of money. The other one is called activation energy. Free energy is the energy that transfers between molecules. So if I lose free energy, which means energy is being absorbed, I typically refer to that type of a reaction as being an endergonic or an endothermic. If I have additional free energy, if I'm releasing energy, I am an exergonic or an exothermic reaction. Inside of all of our cells, molecules are freely moving around, and they're moving around based off of the temperature of the cell. Because the amount of thermic energy in a medium is going to dictate how quickly molecules move. However much additional energy I have to add to the system in order to get those molecules to actually interact with each other is called activation energy. The way in which we measure this energy is typically either through a joule, J-O-U-L-E, capital J, or a calorie. Most of us reference a calorie, but we don't reference the calorie correctly. Everybody looked at their little thing on their cereal box this morning when you poured your Cheerios, or your Wheaties, or your Frosted Flakes, or your Lucky Charms, or whatever else you decided to eat this morning. And on the side, it says there are so many calories within a serving, correct? Correct. They're referencing this number of calories here, what's called a kilocalorie. Kilo always means what? 1,000. And we use that in terms of dietary things because it's easier to use small numbers than it is to use big numbers. Because if it said it had 200 calories, that's really 200,000 calories. A calorie, a little c calorie, is the amount of energy it takes to raise 1 gram of water 1 degree centigrade. A little c calorie, the amount of energy it takes to raise 1 gram of water 1 degree centigrade. Now there's a cool conversion for water because of the specific gravity of water. 1 gram equals 1 milliliter because the specific gravity of water is 1. Specific gravity is simply density. And density is always measured as mass over volume. And so since water's density is 1, mass, volume. So since 1 gram equals 1 milliliter, 1 kilogram will also equal 1 liter. So when we look at the dietary calorie stuff, we're looking at that measure there. When we do chemistry analysis, we look usually at this measurement here. The next thing we have to look at in terms of reactions is the directionality of a reaction. We have what's referred to as unidirectional or bidirectional. Unidirectional simply means I go in one direction and only in one direction. So I either go from A and B interacting with each other to form C, a synthesis reaction, or I go from C breaking down into its components of A and B. Most chemical reactions in the body are enzymatically controlled. Enzymes are biological catalysts. C-A-T-A-L-Y-S-T-S. A catalyst, for those of you that don't remember chemistry, is a part of the reaction that gets used in the reaction but doesn't get used up and doesn't form one of the products. Enzymes are proteins, which means there's a couple of things that are going to affect how reactions occur within the body because enzymes are going to be affected. And one of those things is why we have to worry about getting to this temperature of our body. The opposite of the unidirectional is the bidirectional. The bidirectional is also referred to as an equilibrium reaction. Equilibrium, E-Q-U-I-L-I-B-R-I-U-M. Equilibrium reaction. An equilibrium reaction means that as A and B are forming together, they form C. C will then start breaking down to what? A and B. It can go either direction. The bidirectional reactions are substrate-dependent reactions, which means that if I have a whole bunch of A and B, I'm going to drive the reaction towards what? Towards C. If I have a whole bunch of C, I'm going to drive the reaction towards what? A and B. Everybody follow? The next thing we have to worry about are what are referred to as redox reactions. Oxidation and reduction reactions. Oxidation loses electrons. Reduction gains electrons. But in biochemistry, in our body, in the physiological reactions, we don't really track ions. We don't really track electrons. We track hydrogens. Because hydrogens are easier to measure than electrons in our body. So, in a reduced state, I have additional hydrogens on the molecule. Everybody follow? Because wherever a hydrogen goes, hydrogen has a positive ion charge. What's going to follow it? An electron. So remember, positives will always attract what? Negatives, and electrons are negatively charged. So this is a reduced state because I have hydrogens. This is an oxidized state because I don't have what? Hydrogens. The movement from the oxidized to the reduced state is referred to as a redox. R-E-D-O-X. A redox reaction. Once again, just get the general gist of this, because when we start talking about metabolism, not this term, but next term, a lot of what we talk about in regenerating ATP for our cells is redox reactions. The problem with redox reactions is that electrons may not move as fast as the reaction wants to move. When the electrons don't move as fast as the reaction wants to move, we form specialized molecules that we call radicals. We hear about one type of radical all the time. ROSs, reactive oxidative species. Reactive oxidative species is where I have an element that has an electron spot that's free. When it has an electron spot that's free, it's not happy. And it wants to find its happy place. And the way in which it finds its happy place is it tries to strip electrons from somewhere else. These ROSs go floating through the bloodstream. As they go floating through the bloodstream, they start interacting with the cells of the vessels, and they start latching on. They start latching on, and they basically act like Velcro to the electrons within the other molecules. And what happens to Velcro if we start pulling on it too much? It starts to tear and starts to fray, And in the vasculature, it starts to develop the atherosclerotic tissues.
What does ROS stand for again?
It stands for reactive, oxidative species.
Okay.
So it's an element that's in its oxidative state. It's trying to find an electron because it's short, and it needs to get reduced in order to be stable.
Are those like the free radicals?
Those are the free radicals.
Okay.
ROSs is the free radicals, and that was actually my next point. These ROSs are the free radicals that you hear about all the time. But we usually don't say ROSs because it's one of those phrases that most people don't hear all the time, even though this is what we talk about. Free radicals. In order to neutralize the free radicals, we have to have a molecule that's willing to reduce it. And that's this antioxidant. Antioxidants are naturally occurring within your body. Your mitochondria make them. The mitochondria make them because most of these radicals get formed within which organelle of the cell? Well, it'll take a while to guess. Which one's making all the antioxidants? Mitochondria, so where are these all going to be formed? Mitochondria. You go to the health food store, or you watch the evening news brought to you by your local pharmaceutical company, and they tell you, oh, you need to take all of these extra antioxidants, or these vitamins that have antioxidant properties. Or you have to take this new quote-unquote superfood antioxidant. Just as a side note, there is no superfood. Story for another day. Your body makes enough of these, all by itself. The more active you are, the more likely you are to form these, which means that your mitochondria makes more of these. And if you overconsume these, your mitochondria stops making them. Because it's all about homeostasis, we want to be just right. If I overconsume something that my body is naturally going to produce, my body slows down the rate of that production. And if I slow it down for a long enough period of time, I actually stop producing it. The way in which the reactions take place is based off metabolism. And we talked about this term on Tuesday. Metabolism is the sum of all chemical reactions in the body. They are typically linked together, meaning one reaction links to another reaction. If I need energy in a reaction, I'm going to get that energy from a paired reaction. So, anabolic reactions take small things and make them big. If I'm taking small things and make them big, I have to add energy to it. Because the big things do what? Store energy. And energy can't be created, it has to come from somewhere. So if I'm going to build tissue, I have to get the energy to build a tissue from somewhere else. And that somewhere else is from an exothermic reaction. So an endothermic reaction will always be paired with an exothermic reaction. If I have more exothermic than I have endothermic, then I start getting thermogenesis. When we talk about metabolism, we throw up a whole bunch of words. I like using the word metabolite because it's a molecule inside of metabolism. But the problem is that metabolite can either be a reactant, A plus B, or it can be a product, C. Reactants are the molecules that the enzyme is functioning on to generate a product. The product is the end result. For shorthand, so you don't have to write out reaction, Rxn, is reaction. Shorthand, so you never have to write out function ever again, at least not for me. Just like in math. So, what are the various types of reactions that we have to worry about in terms of the sum total of reactions? Well, the first one is referred to as an anabolic reaction. Anabolism, or anabolic reaction, is the building reaction. In chemistry class, we refer to this type of reaction as a synthesis reaction. What we are synthesizing is a thing known as a mer, M-E-R. If there is just one of these things, we use the prefix mono. Mono means one. So if there's just one of these things, it's a monomer. If there are two of these things, we use the prefix di, and so it becomes a dimer. If there are three or more of these things, we have a poly or a polymer. The mer that we have is going to be dependent upon what actual type of molecule we're looking at. So if it's like sugar, we have a monosaccharide, one sugar, a disaccharide, two sugars, a polysaccharide, three sugars. If it happens to be a fat glycerol, a monoglyceride, one fat chain, a diglyceride, two fat chains, a triglyceride, three fat chains. The way in which we form these bigger mers is by stripping a water from the molecules, which means that we dehydrate the environment. How do we dehydrate it? Well, here is glucose and fructose. These are the mers that form table sugar. The way in which we produce this is sucrase and supersynthase, takes this hydrogen here and this hydroxide here. OH is called hydroxide. We simply just write OH instead of having to write out hydroxide. H plus OH forms H2O. This oxygen doesn't like being single bonded to anything because it has two electrons it wants to pair up with. So what it does is it latches on to that carbon there. And we now have sucrose. The opposite of this is the catabolic reaction. So anabolic is going to build up. Catabolic is going to break apart. So we have our sucrose. We form the sucrose by shedding water from the molecules. We're going to break the sucrose by sticking water in. This type of reaction is in chemistry class called a hydration reaction or a decomposition reaction because the molecule is decomposing. It's breaking into its smaller parts. So, sucrase sticks itself here and it starts kind of wedging the two molecules apart. Water's out in the environment. This oxygen gets sheared away. This hydrogen is going to go to that oxygen, and we get glucose. The OH that's left from the water gets stuck on this ring here, and we have fructose. Your body's constantly doing this with all of the sugars that you're consuming throughout the day. if it's not doing this where it's splitting up the sucrose that you're eating, it's converting sugars that you have in your body into either fructose or glucose. The sugars that your cells use for metabolism to regenerate ATP or glucose-derived reactants and products. The way in which we get this change of molecular shape is through a reaction type known as isomerization. Isomerization simply means I'm changing the arrangement. I'm either going from a cis orientation to a trans orientation. Same side, opposite side. Or I'm moving where a functional group happens to be. In isomerization, we don't change the actual molecule. We simply change how the molecule is arranged. We see a whole bunch of these isomerization reactions in cellular metabolism, particularly in glycolysis, the breakdown of glucose in order to regenerate ATP. The last type of reaction is probably the most important type of reaction in terms of controlling metabolism, and that's referred to as the kination or the kinetization reaction. Kination or kinetization reaction activates. The way in which it activates is it sticks a phosphate onto a molecule. Phosphates are high energy molecules, meaning there's a lot of potential energy there for the cell to use. When we kinase something, we basically transfer energy. So in this case here, we take a phosphate from phosphocreatine and we stick it onto ADP in order to form ATP. Same thing here, we take the phosphate off of PEP, phosphoenolpyruvate, stick it onto the ADP to form ATP. Enzymes do the same thing. We have to energize enzymes in order to get the enzymes to function for most of them. There are some enzymes that just automatically function. There's other ones that we have to stick a little phosphate on. That little bit of phosphate gives the enzyme just enough energy to do this. And by doing that, the enzyme is able to do what the enzyme needs. We can control all of these various types of reactions within our body by controlling various environmental conditions. The principal means of controlling them is by regulating the amount of activation energy that is required for the reaction to take place. The principal way in which we do this is through enzymes. Enzymes that control the rate of a reaction are deemed rate-limiting reactions. The enzymes that control the rate of the reactions are deemed the rate-limiting enzymes of the reactions. So some enzymes just function. Some enzymes are rate-limiting. Remember the difference between the unidirectional and the bidirectional? Unidirectional goes one way, bidirectional goes two ways. Enzymes on the unidirectional steps are rate limiting. They're going to control how quickly I allow the reaction to take place, and they're going to determine how much product I'm allowed to form. The way in which enzymes function is through changing how much additional energy I have to get into the system in order to get the reaction to take place. If I can reduce the activation energy, the molecules will be able to get in better proximity to each other in order to react without me having to jack up how much heat is within the environment.
After a reaction, the enzyme releases the products and can go on to carry out the same reaction again and again.
So, every enzyme will have activation sites, sites where they can bind. The activation sites are chemical class dependent. So we used to think of it as being lock and key, one chemical, one enzyme. But what we found is that it's a whole bunch of chemicals that have the same type of structure for a single enzyme. So what ends up happening is that as the molecules interact with the enzyme is a protein. A little bit of molecular weight causes the protein to change its shape a little bit. When it changes its shape, it either brings the molecules together or it pulls the molecule what? Apart. So if we look at this in terms of the types of reactions that we have, here's our enzyme. Here is our reactant. It comes in, it forms the enzyme complex. In this case here, we're going from one big thing to two small things, so we're undergoing catabolism, a catabolic reaction. These two things cannot fit into that opening anymore. Since they can't fit into that opening anymore, the bond that was formed gets broken. And then they float off. This now goes back to its original shape and is ready to go again. Enzymes will continue as long as they keep their structural integrity. There are some enzymes that wear themselves out relatively quickly. There are some enzymes that go on and on and on and on. And there are some enzymes that we have to constantly produce throughout our lifespan because they wear themselves out so quickly that if we don't replenish them fast enough, we can't do what we need to do. Now we have the anabolic reactions. So instead of having one thing fit in, now we need to have two things fit in. So the two things fit in, they form the reactant complex. The enzyme moves the substances close together. This can no longer fit into the enzyme. It kicks it out, and then it goes back to its original shape, ready to go again. Isomerization reactions work the same way. We have the reactant that fits in. The enzyme then kind of stretches it. You guys have played with silly putty before, right? You have the ball of silly putty. Try to stretch it out. You can't put it back into the egg without... Same thing's happening here. So we interact with the enzyme kind of pulls and kinks the molecule. The molecule no longer has the correct shape to fit in the enzyme. It gets dissociating, goes and does what the product has to do. The last type of reaction is that kinase reaction, or kyanization reaction. So, here is the thing we have to activate. Here is the enzyme that does the activation. So the enzyme has a phosphate on it. What the enzyme does is it transfers the phosphate onto the reactant. We now have an activated product. The different types of reactions get identified by the different types of enzymes. All enzymes have the end phrase ASE. All enzymes have the end phrase ASE. So, if I have an enzyme that is known as an isomerase, that's this enzyme. An isomerase does isomerization. If I have an enzyme that is a kinase, it does kination reaction. If I have an enzyme that is a synthase, if I have a synthase, I am going to synthesize. Follow? So what's going to affect how quickly the reactions take place or how well the enzymes work? Well, the first thing it's going to affect is how much stuff do I have? Do I have enough of what I'm working on? Have I made too much product? Negative feedback. If I make too much product, I'm going to slow down how much more product I'm going to make. If I don't have enough raw material, am I going to be able to make the product? Because enzymes are proteins, they are sensitive to environmental conditions. In particular, pH and temperature. Proteins do what's known as denature in changes of acidic condition. So proteins are usually kind of folded and woven around each other. When they get exposed to different types of pH, they either clump down or they open up, depending on if we're going acidic or we're going alkalinic. Same thing happens with temperature. If I elevate the temperature of a protein, the protein unwinds. Anybody eat fried eggs? Why is the egg white, which is principally albumin protein, go from being clear to being white? It goes from being clear to being white because it goes from having all of the proteins lay out like this to all the proteins laying like this. So you can see through it to not seeing through it. It has to do with the proteins unwinding. You can do the same type of thing with eggs if you dump them into vinegar. Take an egg, dump it into vinegar, same thing happens where the egg white actually turns from clear to white. The other thing that's going to impact this is a thing known as competition. So, the binding sites fit for a class of chemicals, not a single chemical, which means a whole bunch of things can fit into it. Most of the competition is referred to as inhibitive competition, where we have things that are competing for the same spot. So, the green guy wants to get in there. But if the pink guy gets in there before the green guy does, the enzyme cannot function on the green. That is inhibitive competition, or competition inhibition. The other thing is sometimes enzymes sit in idle. They need to be turned on. Or they're sitting at red line and they need to be turned off. That's what's referred to as allosteric regulation. Allosteric is the big fancy term for shape of the enzyme. And by changing what is in the enzyme and what's referred to as the regulatory site, we go from having an active enzyme to an inactive enzyme. So, we're going to inhibit this guy. This guy's open, he can go ahead and do it. Here comes an inhibitor. As it fits in, it changes the shape